which in contrast to single particle tracking methods where you follow single particles across the nucleus and their trajectories, allows you map the motion of the chromatin here across the entire nucleus in real time. Now, what we use for that, it's a staining using H2P GFP histones for those of you interested. And what this method allows us to do, not only we track the motion across the entire nucleus, but we actually can vary the time scales at which we map it. So we can vary the interrogation window, which gives the method its kind of spectroscopy nature and allows us to map basically the dynamics on different time scales, as here short and here long time scale. So what we found with that was this very striking result that at short times, the motion of chromatin is truly uncorrelated. It seems to randomly jiggle, where at long times we start to see these regions of coherent motion. So my vectors are here color-coded by direction. So you start to see these patches of same direction, which I have here outlined by this picture. And as Peter has already shown, by calculating the spatial autocorrelation function, we can actually detect the size of these regions of coherent motion in different time scales. So we have asked ourselves, obviously, what do these regions mean? Where do they come from? What are they? So when you look at this picture, that reminds you immediately of a similar picture that we have seen in this conference and in the workshop already a few times. And that is the beautiful fish image of chromosome territories. Now you can see that at the order, the length scale of the coherent regions and the territories is about the same. So it was not so outlandish to ask the questions, are we actually seeing the chromosome territories moving? So we've done an experiment where, in addition to our H2B GFP, from which we obtain our DCS signal, we have added second marker that now has stained only half of the chromosomes red. That allows us basically simultaneously to measure from the green signal our DCS map, so those are the arrows here in this image, and from the red signal that corresponds here to the gray mask in this image, and that provides you here the boundaries between the gray and non-gray, or here red and non-red regions, give you physical boundaries of chromosome territories. So you can analyze this type of data and overlay basically this dynamic information with the static information and look where the territories sit. So we find in some cases, certainly as here, you have here the gray region, white region, gray region, so this is the blue line is the boundary. We find that these pass each other, so two territories definitely passing each other. That would be here basically green chromosome passing the blue one. However, this is very rare. In most cases, what we find is that you have two neighboring territories, as here the gray and the white one, and the coherent region actually expands beyond the boundary, right? So it actually spans parts of two chromosomes, as shown by this cartoon, where you see the parts of blue and green territory move in this box together coherently. So this kind of suggested that, okay, we don't see the motion of the entities themselves. It might be something functional. In the school on Sunday, I have explained to students all the possibilities that we have explored so far, specifically to molecular motors. Here, I'd like to present you more the physics picture that we have developed from these observations. So the first questions we have asked are, is this coherent motion ATP dependent? Is it active or not? So we have depleted ATP in our cells and first looked at the mean square network displacement, which is an equivalent of mean square displacement that we can calculate from our data. And first, when you compare just this displacement, you see that this is how it looks in a healthy cell. So the red curve, where after you deplete ATP, the dynamics is reduced to certain levels. So all this difference basically correspond to ATP-driven processes. Moreover, what we find is the coherent motion here disappears. So moreover, what we can do, we can calculate the power spectral density of our fields. And now what I'm showing here is the calculation for both the wild type, so this is the control cell, and the ATP depleted. And now, again, I have the colors corresponds to different time scales, and the solid lines are the healthy cell, the dashed lines are the ATP depleted. So what you can see that at all time scales, these curves are very similar, except for here, this large wavelengths, low Qs, where we have a strong deviation. So this suggests very nicely that active events are highly influencing the large wavelength fluctuations. So we've moved further on that and started to think what type of active processes can be actually contributing. And from a physicist perspective, obviously, we did not go one by one, 4,500 nuclear ATPases at a time, but we categorized them into two big categories. And so we have a look, we have defined one category as vector events. Those would be the molecular motors that act on chromatin fiber locally in a way which has a direction as well as magnitude, so they can be described by force dipoles, as you can see here, and the list of motors that could be approximately described by that. And then, of course, scalar events, which would not have a direction, yet they would have magnitude. So you have in the nucleus a lot of condensation and decondensation events. So these sources and sinks of concentration fluctuations could be described by that. For example, the chromatin remodelers actions. And so together with my collaborators, Shura Grossberg right here, Robin Brunsma, and Yitzhak Rabin, we have developed a theory based on our experimental data which provides a hydrodynamic description of the polymer solution or of the chromatin solution inside of nucleus, starting with a two-fluid model where the polymer is the chromatin and the solvent nucleoplasm. You start with an equilibrium solution, so Shura has shown it yesterday in much greater detail, and on the top of this equilibrium solution you put the active perturbations that push you out of equilibrium, so for both the vector events and scalar events, so force dipoles and condensation, decondensation. Yesterday, Shura mentioned that we could go even further, and the activity in the nucleoplasm might have been could be considered here as well. So I just want to mention here the results of these theories or the predictions. This theory shows that the large wavelength fluctuations, so the coherent motion that we see, are dominated by the vector events, so by force dipoles, whereas the small wavelength fluctuations are dominated by the concentration fluctuations, so those scalar events. So that has motivated our further efforts, since when you think about it, how can a single force dipole, so single molecular motor, actually cause a coherent motion across a few microns, right? So our regions are 3 to 5 microns. One motor is 3 to 5 nanometers. So what we propose, or what the hypothesis is, that one way to do so would be if those force dipoles would actually nematically align. Now, how could that in reality happen? There are two ways how you could go about it. Either you could have a local transient force that would be applied locally and thus locally here align you these guys, or we have to consider we are here at a regime of very low Reynolds numbers, and thus the hydrodynamic coupling is very strong. So we started to explore this both experimentally as well as with simulations, kind of to look into what role the hydrodynamic coupling plays. And specifically the simulation, we started to collaborate with Mike Shelley at Courant at NYU and David Santillan at UCSD. And this is here one of our very first movies where you can see a in silico nucleus where you have 4 chromosomes, 4 chains swimming around. So which are interacting via nucleoplasm. So basically you don't see the solvent, but there is a physically solvent. And these chains are basically here, in this case, semi-flexible polymers or flexible polymers with actually sites of activity. And this activity is modeled by the force dipoles that are acting on the chain and on the fluid. So they generate their local flows which interact, and the whole interaction here between the chains is via hydrodynamics. So here just a brief description, but due to time I won't go into detail, but basically the numerical simulation solved a Langevin equation for every beat where all the forces considered are being involved, so from the thermal fluctuations to excluded volume to the active forces on the links which are here in red. That's basically where our activity is. And then also locally, basically at every point, then the Stokes equation is being solved for the local flow caused by such a force dipole. So here, just to give you a little intuition for how such behavior looks like, if you would have just a passive Brownian chain in unconfined case, we know what to expect. If you put contractile dipoles on your chain, then it behaves a little bit as a accelerated thermal Brownian chain. However, when you put extensile dipole, you see that over time, basically the chain has a tendency to stretch and to achieve its largest extension. Now, when you put a long polymer now into a confinement and apply these type of dipoles here as well, what you see that here, so this is the Brownian motion. For contractile dipoles, we don't see big differences. We see it's more kind of accelerated Brownian motion. But for the extensile dipoles, we start to see small regions of aligned of our toy chromatin fiber. So we can do a direct comparison with the data. So this is what I have shown you, before, our ATP depleted, our passive chromatin dynamics, the active control state, the DCS data that we collect, and this is the correlation function that we measure. So the red curves correspond to the correlation function for these different time scales, where here now, instead of colors, I have different shades of red. So you see coherent motion for the wild type, whereas for the ATP depleted, those are the blue curves that are all collapsed on each other. Now, when we look at our different chains, what they do inside of confinement, you see that passive and contractile exhibit completely decorrelated displacements, but for extensile dipoles, we start to observe these regions, similarly as in wild type, we start to observe these regions of coherent motion, and we can also recapitulate basically this behavior qualitatively via a correlation function. So we have, of course, explored the whole phase diagram. What is kind of the ratio? How many of the dipoles do you need? How strong do they need to be? And what this shows you is a phase diagram here, which shows the strength of a force dipole versus kind of the number of the dipoles acting. And you find that there is a beautiful boundary. This is the region of coherent motion. So kind of few very strong ones can achieve the same effect as many weak ones. So what this shows us, that this is, of course, only a toy model of chromatin that is purely to explore what hydrodynamic interaction could do, and it shows that the hydrodynamic interaction alone could indeed lead to large-scale coherent motions that we've observed before. So how many minutes do I have? 5. At least, yeah. Yes. So, so with this, right, I hope to have convinced you that this is not elf-stirred, but self-stirred. And this obviously will have major implications for the genome organization as well as gene regulation. So what we are very much after and would love to do is obviously to reconcile this dynamic picture with this beautiful static detail that we have from high C, that we have learned from RS and others, right? That ideally, hopefully, in the years to come, our two worlds converge and we can identify features moving inside of living nucleus. So with that, I'd like to move to the second part a little bit, and that's the rheology. Since this is a beautiful nucleus, here it's a beautiful example or canonical example of a confined polymer which is alive and which is active. So we want to learn about its rheology. The classic approach is to inject particles, magnetic, non-magnetic, either observe their motion, apply forces, and so on. So we have been injecting particles ourselves, and one thing that we notice, the cells tend to suffer when you use injection needles, since you cause a big hole. So we were trying to figure out an alternate way to go about it, and realized like, well, chromatin solution is actually filled with number of subnuclear bodies, and we're learning about new ones every day. Why don't we use some of these little structures that are already present? So specifically here, we saw that even at interface, the dynamic is very rich. And what is the interface of the nucleolus, which is the largest structure? And as we know from a few days of this meeting, basically the archetype of the liquid condensates currently in cell biology. So we decided to use nucleolus as a rheological probe and try to learn by observing nucleolus about chromatin around it. So the first thing that we have done, we have observed the surface fluctuation of the nucleolus with the high spatial and temporal resolution, and we found that the surface indeed fluctuates. It flickers very fast. Those are very, very small fluctuations, but very nicely visible. And from those, here you can visualize them. So those are now the fluctuations here in polar coordinates. This observation, of course, is very much consistent with nucleolus being a liquid droplet, because if it would be a solid particle, its surface would not fluctuate. So from this, we can also estimate the surface tension of the interface, nucleolus and chromatin solution. Of course, this is just the upper bound when we assume it's thermally driven, and we have an estimate of surface tension to be at the order of 10 to minus 6 Newton per meter. So this is extremely small. It's, however, comparable to the polymer colloidal solutions. It's also very similar to the frog nucleoli that have been studied earlier by Cliff Brangwyn. So furthermore, what we have done, we started to, or attempted to, look at the fusion of human nucleoli. As we know, a fusion of two particles or liquid condensates, right, that started to be kind of a canonical test for the liquid condensates. Are they indeed liquid? But also, we realized that this can be very informative about the physics of the chromatin as such. Now, to find actually fusion of nucleoli inside of human cells is not easy. Indeed, it's very tricky. So such an image has been shown about 60 years ago for the first time, where people have caught kind of the nucleoli in the act, but they could not tell, is it a fusion of solid particles or coalescence of liquid droplets? So in order to distinguish between these processes, you have to catch dynamic data. So you really have to see how the shape changes, how the neck in this dumbbell shape, how it grows. And that's where the information lies. But there is a reason why it's very difficult here, because in contrast to frog eggs, where you have 2,000 nucleoli, we only have 2-3. And in contrast to frog nucleoli, which are at the order of 20 microns, our are 1 micron small. So it's just the art of kind of being at the right place at the right time, so you catch the process, and you don't know when and where it occurs. So here, for that, Christina Kargin, a grad student in my lab, sitting right there, she has developed a beautiful semi-automated algorithm that allowed her to go to 10,000 cells that she inspected, and from those she identified 150 fusion events, from which 14 were captured in XY plane, where we have the highest optical resolution and therefore the biggest detail of information on this neck, which holds the whole information, what forces are participating in this fusion process. So if it's a liquid droplet, then obviously the fusion is then driven by surface tension. So your droplet, the final droplet wants to be spherical. So that means that the capillary forces are pushing it to be round, yet it is being resisted by either viscous or inertial forces based on the system. This system obviously is highly viscous, as I'll talk in a minute. So here we will be discussing viscous ones. So the information, as I said, is in the neck. So you need a detailed information on the growth of the neck as a function of time. And this is exactly here what Christina is doing here in this experiment. So you see she's simultaneously collecting the green signal from the chromatin and the red signal from nucleoli. And so here, this is the neck, which is here being plotted as a function of time. So here are the beautiful 14 coalescences or 14 fusion events that we were able to catch. So those are the only ones currently existing in this world. So we were very proud of it. And it took us a long time, and it took Christina a lot of work to get it. And this data is highly informative. So first of all, when we evaluate, as I told you, what type of regime is this situation happening, the Reynolds number here is extremely low, telling you that it's the viscous forces that dominate this coalescence. That allows us to go even further and look at the temporal dependence of the behavior. How does it change if the outer fluid would be dominating or the inner fluid? So the temporal dependence for the growth of the neck is very different. If the nucleoplasm, so the outside, would be dominating, then it should go as a T to 1/2. If it's the inner fluid, then it should go linearly with time. The difference here is exactly as if you take two water droplets, and this is the situation if these two water droplets would coalesce in air, so they go fast together. Whereas this situation is a situation where you have two water droplets coalescing in honey, in a thick honey. So you could imagine it will take them a while, even though they're able to go much faster in other environment. So we have, within these theories, we have rescaled our data with respect as all the nuclei, they varied slightly in size. Also our experiments, the way how Christina catches the fusion event, there's a slight time shift at the beginning. So correcting for all of that, we find the data beautifully collapse on a power law of T to 1/2, which basically clearly shows here that it's the chromatin, the outside fluid, that actually provides resistance to the coalescence of those two nuclei. And it's the chromatin that basically dictates the time scale of this process. So from this, obviously, so we have further, we have then use theories developed by Sid Nagel and colleagues, where we can go further and obtain actually here even the ratio of the surface tension and the viscosity of the surrounding fluid. And this is this ratio as measured from our data. Now, don't forget, we have already measured surface tension in a separate independent experiment, so we already have gamma. So therefore, now from this ratio, we can also determine the viscosity. So with this, basically what I wanted to show you is that purely by looking at the cells, not touching them, not perturbing them in any way, this is purely by looking, we have completed a complete rheological characterization of the chromatin solution and obtained its viscosity and the surface tension of the nuclear chromatin interface. So with that, I'd like to just conclude on a couple of thoughts. Why would a cell want to slow down this kinetics of coalescence so much? So for that, what we have thought and what we speculate, the following might be relevant. You have to think, what is the nucleolus actually doing? So it is a site of ribosomal biogenesis. For that, it actually does the transcription of rDNA of the ribosomal genes that have an average length of 7 kbp. Now, at this given transcription rate, it would take about at the order of 100 seconds to transcribe one of those genes. So if the coalescence times would be, as for example, in vitro for purified nuclei in aqueous buffer is seen in a couple seconds, then you can imagine those processes at 100 seconds would be highly perturbed, right? It would be like an earthquake for the transcription. However, if you slow down the coalescence here 1 order of magnitude, then actually the transcription could be happening unperturbed, and the coalescence can happen this way. This is actually, by the way, this speculation is supported by another observation by an earlier study, which has shown that at the moment when you stop the rDNA transcription inside of nucleolus, nucleolus dissolves. So they have to be active in order to be alive, so to say. So this is our speculation here that the long coalescence times might be needed for unperturbed transcription of our DNA. So with that, here are my conclusions, but I might be out of time. So I just go ahead and thank my team, since they have been instrumental in all of these efforts. And here, of course, I'd like to thank also my collaborators, the funding agencies, especially NSF and Physics of Living Systems, and you for your attention. Thank you. And before I take questions, just one thing I wanted to do, very short, 10 seconds advertisement. In a year from now, Shura Grossberg, Job Decker, Ralph Evers, and myself are organizing a conference at the Kavli Institute of Theoretical Physics in Santa Barbara in California, Chromosomes, Organization, Structure, and Function. Please take a note if you're interested. We'll be happy to have you.
All right. So Irene, I think you were first.
Beautiful talk, thank you.
Thank you.
So I have a question to the first part, because you mentioned that you have this toy model. And I'm, as a biologist, also would like to play with your toys. But I have to disagree with the design of this model, because what you show, all your polymers are equally mobile.
Yes, yes.
Independently whether they are at the periphery of the nucleus of the model or inside. I think this is that model for biologists, because we know that chromosomes are anchored to the nuclear lamina very firmly. They don't move.
We are doing that, yes. So this is the version 1.0. Currently, we have already incorporated, so the model is basically being decorated as a Christmas tree now, and other things are being added. We're adding the links to the envelope. We're adding the heterogeneity. We're adding different stiffnesses to the links and so on. Yes, yes, yes.
So it's not a toy anymore.
No, no, no. But this was basically purely, right, if we just look at one kind of interaction, can we see effect?
And the second is not a question, but it concerns the second part. Actually, there is a very easy way to observe a lot of merging nucleoli. It's known that the nucleoli start to fuse towards the G2. Synchronize your cells, and you have hundreds of the events.
Well, actually, so we've tried several tricks. So we found, so Christina can tell you at her posters tomorrow more. That's what we taught from literature as well. We find actually they fuse at a variety of times. Yeah.
Okay. Yes, one last. One last question from Mr. Decker.
Yeah, hi. Wonderful talk. And about the first part, when I saw that model, you mentioned that the slow dynamics is driven by ATP-dependent processes. These are micron large rearrangements. I was thinking SMC proteins are the prime candidate for that, but they were not on your list.
Which proteins?
SMC, structural maintenance chromosome protein,
yes,
cohesion condensing.
Yes.
Isn't that a logical candidate to drive such large scale
Certainly,
structures?
certainly. So, I mean, I have listed there a couple of examples, but that's just basically to visualize how you can think about the vector versus scalar, but not, this is not a list that is limited to those.
Because you mentioned, like, even in your phase diagram, like single entities which have large scale rearrangements could do the thing, right?
SMCs are a fantastic idea, and we are indeed thinking about it also how to add some specific signatures for them, how they could be actually, you know, linking neighbors and so on. Yeah.
All right, so let's thank Alexandra one more time.